So thanks a lot for inviting me to speak here today for what I hope will be a lively discussion around human pluripotent stem cell models in the era of big data. So please feel free to interrupt with questions, with comments at any time. I really mean for this to be a discussion. So I'll start with a brief introduction about stem cells and their history. And then I'll highlight some of the promises, but also some of the current limitations of stem cell models. And finally, I'll discuss the path forward, or how do we go from there in the field. And then I'll hand it over to Matt Tegtmeyer in my group, who's going to tell you about the stem cell repository that we have established here at the Stanley Center, as well as about some exciting collaboration that we have going on that utilize this resource. So human development starts with a single cell, the fertilized egg, which then divides and gives rise to two daughter cells, which then divide again, and so on, until a blastocyst is formed and eventually a fetus. So there are many steps that are needed to form an adult body and even a baby. And along the way, many different types of cells need to be made. Stem cells are different from other cells in the body because they can both self-renew, so make copies of themselves, but they can also differentiate, so give rise to specialized cells or differentiated cells such as muscle cells, skin cells, neuronal cells, basically any cell type in the body. The two types of human pluripotent stem cells that are typically grown in the lab are embryonic stem cells and induced pluripotent stem cells. Embryonic stem cells are taken from inside the blastocyst, which is a very early stage embryo. The blastocyst is a ball of about 50 to 100 cells, and that is not yet implanted in the womb. It is made of an outer layer of cell, the trophectoderm, and a fluid-filled cavity, as well as many cells, like 50 to 100 cells here in the middle. Embryonic stem cells are found in this inner cell mass right here. They can be taken from the ICM and then cultured and expanded in the lab. The first mouse embryonic stem cells were derived from blastocysts in 1981 by Martin Evans, Matthew Kaufman, and Gail Martin, but it's not until 17 years later that they were first derived from human blastocysts in 1998. Induced pluripotent stem cells, on the other hand, are derived from somatic cells, such as blood or fibroblasts, and they can be reprogrammed to an embryonic-like pluripotent state, typically by the addition of transcription factors such as OCT4, KLF4, SOX2, and c-MYC. The first human-induced pluripotent stem cells were generated from mouse fibroblasts using retrovirus in 2006 by Takahashi and Yamanaka, and then from human fibroblasts a year later by the same group. And then they were first used for disease modeling in 2008 and 2009 by the groups of Dimos and Egan and Lee and Studer. So while non-human models will continue to provide essential biological insights, there are aspects of human biology that are most accurately captured using human material. For example, disease-associated regulatory variants are often not well conserved, which necessitates access to human cell types. Additionally, there are many evolutionary differences between brain structures in animals. For example, the human prefrontal cortex that is associated with high-order cognition and function is not well conserved across species, and it has been implicated in neuropsychiatric disease pathology. However, we have limited access to tissue from the developing human brain. For this reason, we have turned to the promise of stem cell biology. So human pluripotent stem cells enable the development of an unlimited source of many types of human cells that can then be used for basic research or even therapeutic applications. New protocol for differentiation give us the ability to generate defined and purified cell types that are relevant to disease. And perhaps most importantly, we are able to generate these cell types from human patients, giving us access to patient tissues. Many neuronal differentiation approaches have been developed that are relevant to neurodevelopmental, neurodegenerative, and neuropsychiatric diseases, such as cortical excitatory neurons, inhibitory neurons, or even astrocytes. These approaches include ways to grow neurons in two-dimensional models, where typically they form connections or synapses and neuronal networks. One of these approaches that is widely adopted is the directed differentiation approach that includes the addition of developmental patterning, which consists of pharmacologically inhibiting or activating various molecular signaling pathways that have been linked to brain development. These approaches typically give rise to mixed populations of cells, of neuronal cells. The other approach that is commonly used to generate neurons in monolayer models or two-dimensional models is the transcription factor reprogramming approach, such as overexpression of the neurogenin 2 transcription factor. This is a really very strong and powerful neuronal differentiation factor that often gives rise to pure populations of neurons, often as single cell types. More recently, ways to grow neurons in three-dimensional models have been established to recapitulate early brain development. These are collectively known as organoids, and the two main ways to generate organoids include either the use of developmental patterning or no patterning. If no patterning molecules are being used, typically this generates different cell types and also different brain structures, as we have heard extensively from the groups of Lee Rubin and Paula Arlotta and Silvia Velasco. Beyond the promises of human pluripotent stem cells, I would like for the remainder of my talk to discuss important considerations to keep in mind while designing human pluripotent stem cell studies. And here I want to specifically acknowledge Lindsay Barrett because the two of us have extensively discussed these considerations and summed up the key concepts in a review format. So today I would like to highlight the sources of variance in human pluripotent stem cells. I'll also discuss the issue of reproducibility or scale. And then I'll talk about in vivo fidelity, or how closely do these in vitro cell types resemble the cells found in the human brain. And I'll conclude with some regulatory considerations. So there are many factors that could impact the presentation of cellular phenotypes in vitro, regardless of the genetic basis of disease. These include differences in human pluripotent stem cell derivation, whether we're using embryonic or induced pluripotent stem cells, which method has been used to reprogram the induced pluripotent stem cells, the donor cell type, and the donor age. Many genetic or even epigenetic variation can arise over time in culture, such as trisomies, copy number variants, and X chromosome status in the female lines, which I'll discuss some more. And human genetic variation can also influence phenotype expression. It is important, for example, whether we're using related or unrelated control and which sex is the cell lines. Another important consideration are the cell types that are present in the culture and in what ratios. And of course, which differentiation scheme is being used, monolayer versus an organoid approach, if developmental patterning is being used, and if transcription factor overexpression is also being used. And finally, which time point is chosen for analysis, whether it's the pluripotent state, the progenitor state, or post-mitotic cell states. So as I mentioned, in addition to genetic alterations, epigenetic alterations can also arise over time in culture. One important example is X chromosome inactivation. While female cells in vivo normally have one active X chromosome and one inactive chromosome, female human pluripotent stem cells over time in culture can show erosion of X chromosome inactivation, giving rise to cultures with either two active X chromosomes or a mix of active and inactive chromosomes. Obviously, for this reason, the sex of human pluripotent stem cell line is an important consideration of studies of X-linked neurodevelopmental diseases. And the lack of ability to readily monitor or manipulate X chromosome inactivation in vitro may contribute to the preferential use of male lines. For example, out of 8 different studies using a total of 39 induced pluripotent stem cell lines to model autism spectrum disorders recently, none of them were female, they were all male. And while there's a strong scientific reason for doing so, it is important to keep in mind that a failure to study female lines may limit our ability to understand how sex differences intersect with disease liability. So there's therefore a need to develop novel methods to monitor and manipulate X chromosome inactivation in vitro. Adding to the complexity, it has become clear that both human genetic studies and induced pluripotent stem cell resources have a diversity problem, specifically with regard to the overrepresentation of European ancestries. We have heard extensively from Alicia Martner about this. So while individuals of European descent make up 16% of the worldwide population, they account for 79% of participants in genome-wide association studies. As Alicia has shown, this disparity translates into lower genetic prediction accuracy in non-European populations. Unfortunately, human-induced pluripotent stem cell collections that are available through repositories in the United States and Europe also have a similar bias in the stem cell collections. We have looked here at the collections that are present in the US, such as CERM, Coriell, NIMH, and NINDS collections. And we have found that they have between 71 and 82% representation of individuals from Caucasian ancestries. The picture is not much better in Europe, where the Hipsky collection, which is the largest in the UK, has 91% representation of individuals from European ancestry. As human genetic data sets are expanding to achieve greater genetic diversity through efforts like the Stanley Global Neuropsychiatric Genetic Initiative, as well as the All of Us research program funded by the NIH, therefore, it is critical for IPS collections to also similarly diversify, to capitalize on the functional assessment of variants that are identified in non-European ancestries. Next, I'd like to discuss the design of appropriately scaled studies, or in other words, how can we get reliable and reproducible results? So some statistical power calculations have fueled large-scale genetic studies, and which have been very successful because of this reason. However, experimental biological studies largely lack statistically informed standards of how many cases and controls are needed to draw meaningful conclusions given specific underlying genetic architectures. A good practice for study design is to first run power calculations based on a pilot experiment, and then compute the variance in a specific experimental setting. The needed sample size can then be estimated based on the amplitude of the desired effect. So the larger the variance, the bigger the sample size is going to need to be. A recent study of sensory neurons derived from 100 healthy induced pluripotent stem cell donors calculated a between sample variance that was larger in these stem cell-derived neurons as compared to the dorsal root ganglion neurons, shown here in yellow, which led the authors to say that gene expression is more variable in vitro cultures as compared to the relevant primary tissues. They further determined that the larger proportion of the variance was explained by the differentiation batch, the neuronal differentiation batch, closely followed by the donor IPS line of origin, so the genetic makeup of each individual line. This, of course, will vary, so the better and more reproducible the neuronal differentiation protocol is, the smaller the variance between batches. Coming back to the question of how to get reliable and reproducible results, or in other words, how can we maximize the statistical power? One can decrease the variance in a study by using related controls, patients with shared genetic variants, or neuronal protocols that have less cellular heterogeneity. And finally, one can pool samples together for processing, as we have heard from Kevin Egan and Steve McKell's group, and that Matt will allude to in his talk as well. Whenever possible, and I know this is costly, but when possible, it really pays off to increase the sample size, so increase the number of biological replicates, and also to validate the findings from these patient-based cohorts using isogenic cohorts, so introduce a specific variant of interest in an isogenic background using genome engineering techniques such as CRISPR-Cas9, and then to be able to really validate the causality of the results. Another important consideration in human pluripotent stem cell studies is in vivo fidelity, so how closely these stem cell models resemble the cells that are found in the in vivo human brain. And there are indeed key differences between these in vitro culture system and the in vivo scenario. These include cell types that are present in different ratios in the dish, the maturity of the cell types that are derived, so often neurons derived from stem cells, for example, correlate most closely to fetal stages of human brain development as opposed to adult stages. Many brain structures or features are missing, such as vasculature, myelination, and microglia. Also, a recent study showed that cells and organoids can display high levels of metabolic stress. And finally, lamination, which is the organization of cells and cortical layers, is not well conserved in vitro either. Despite these shortcomings, we should keep in mind that there are many ongoing innovations that hold great promise for the improvement of these in vitro systems. For example, the missing cells, such as microglia or even endothelial cells, can be derived separately and then added back to the cultures in the dish. Also, bioengineering approaches could be used to generate scaffolds that can then be used to separate cells into appropriate layers. And finally, many new media preparations are coming out that hold great promise to improve maturity in the dish. While we're talking about this issue of in vivo fidelity, I want to make here an important point that this is really a very, very important aspect of the debate here and consideration while designing studies, but it still has many open questions. And this is because there still are critical gaps in our knowledge of the human brain today. For example, do we know which features are required to call a cell an inhibitory neuron, an excitatory neuron, or an astrocyte, in vitro and in vivo? Moreover, how many subclassifications of each major brain cell type exist, and what are their key molecular and functional distinctions? And do we even have the resolution to assess which features are present or absent at each cell state? And most importantly, to what extent does an experimental system need to match the human in vivo setting in order to facilitate important biological insight? So resolving some of these questions will require the acquisition of more complete data sets that go beyond the current transcriptional and epigenetic profiles that currently exist to incorporate proteomic, morphologic, and functional metrics. And then we can really compare these data sets, these gold standard in vivo data sets, to the cells that are derived in the dish, and evaluate the utility of specific differentiation paradigms. Last but not least, regulatory considerations. A majority of the early human pluripotent stem cell derivation efforts were taken on by individual laboratories. These initial collections often had restrictions in the consent forms from the donors, which limited their use and whether these cell lines and data sets could be shared or deposited in the public domain. Investigators today must meet requirements from funding agencies and governmental agencies, as well as scientific journals, to deposit data into accessible repositories. And projects increasingly involve international multi-center collaboration to meet cohort diversity ideals, as well as requirements from statistical power calculations. At the same time, compliance and regulatory frameworks limit the ability to share data and cell lines to be consistent with donor consent. This, in turn, can hinder publications, and most importantly, it can preclude the wide use and distribution of these cell lines and the derived data. Luckily, some newer human pluripotent stem cell collections are striving to simplify consent forms and maximize the ability to share and disseminate data and reagents, which means that in the years to come, we hope that lines and data sets will be more easily shareable and more widely usable. So looking forward, we would like to advocate for a conscious approach to sex and ancestry in human pluripotent stem cell studies, for power calculations to estimate scale and lead to reproducible and reliable results, and finally for the investment in human fetal tissue research and technologies to improve the fidelity of these in vitro derived cell types, as well as for greater access to human pluripotent stem cell collections that are broadly consented for distribution and data sharing, such as phenotyping and sequencing data. So to conclude, I would argue that by using human genotypes that contain relevant molecular machinery, we can position ourselves to make discoveries with relevance to human disease. Human pluripotent stem cell resources are an important partner alongside human primary tissue and in vivo animal models to move from psychiatric genetics into biological insight and actionable clinical information. So with this, I would like to hand it over to Matt Tegtmeyer in my group, who will tell you more about the stem cell repository that we have established here at the Stanley Center. And I'm happy to answer any questions, meanwhile, if you have questions.
Great. OK, so Ralda gave you a good overview of the history, promises, and pitfalls of the use of pluripotent stem cells for modeling and studying human diseases. And I'm going to give you a broad overview of the stem cell resource we've been building here at the Stanley Center over the last few years, how we're sort of trying to address some of the pitfalls that Ralda highlighted, and additionally how it's currently being used to fuel science and technology being done here at the Broad and our affiliated schools. So the primary goal of our resource is to provide the Stanley Center community with not just a cellular resource, but also a genetic resource that will help to enable us to model neuropsychiatric disorders. And as a brief organizational overview, this sort of begins with a sample collection, which happens from our clinical providers and partners, both here, local community in Boston, wider US, as well as all over the world, and I'll talk about that in a little bit. These samples that they retrieve from patients, is usually in the form of a fibroblast or a blood sample, will come to us first, and then we send these off to reprogramming facilities. And generally this is either at the New York Stem Cell Foundation in Manhattan or at the IPS Reprogramming Core at the Harvard Stem Cell Institute up the road. And then another large subset of our collection are lines that we have purchased from CDI and Fujifilm in a partnership with CERM. And these are already pre-reprogrammed, so they come to us as IPS cells. Once they come to the Stanley Center repository, they go through a very standard expansion and banking pipeline. Every line undergoes the same expansion procedures, and then they're all submitted for genotyping and sequencing. And I'll sort of talk about what we do with this information later. To date, over the last 2 years, we have now built a resource that has over 670 pluripotent stem cell lines. And as Ralda sort of highlighted some of the current well-established collections here abroad, or here within the US and abroad, you can see in a short time we've really created a large resource that rivals some of the ones that have been multiple year efforts. So we do have a vast resource here for collaborators to utilize. And the future is both bright and busy. We have over 10,000 people consented for stem cell derivation through partnerships throughout the world. Thanks in large effort to a lot of the people here at the Stanley Center. The collection so far and in the future has really been a global effort, and we continual efforts to try to grab samples from collections where we're missing certain ancestries to try to create a more equitable cellular resource. You can see represented on this plot, dots that are in red are representative of locations where cell lines we already have in-house and are currently being used have come from. Green are cell lines where samples are being collected now from our clinical partners. And then blue are regions where collections are becoming established and efforts are being focused to sort of harbor samples. You can see from Africa and South and East Asia, areas where we're really lacking currently. We have in our collection a very broad diagnosis of different neuropsychiatric disorders, as well as age and ancestry match controls and a lot of family cohorts, which I'll talk about in a minute. And you can see we have a large subset of intellectual disability, schizophrenia and its associated subtypes, schizoaffective disorder and others, ASD, and a few other non-psychiatric controls, so heart disease and diabetes that might have some comorbidity. As Ralda had alluded to, the genetic disparity, there's also sort of a cellular resource genetic disparity that we have, although we have efforts aimed at reducing this. So our current cohort has about a 64% representation of Caucasian, and we have a large subset that are unknown or other undisclosed. So these are just non-reported from the providers, or people were particularly not sure about their ancestry. So if we actually thanks to the help of Dia and Stephen McCarroll's group, if we actually plot sort of the genetic data from our samples using the 1,000 genomes as a reference, you can see the majority of our samples are still sitting in the European ancestry with a few admixed populations, and then very limited in the South Asian and African cohorts. But we are primarily focusing future collections to acquire samples from these underrepresented ancestries, and those are sort of getting underway and establishing that direction now. So hopefully as the years come, we will be able to really come down to these two little islands and build a bridge in our genetic collection. As Ralda alluded to, the lack of sort of sex equity in stem cell research, especially for neuropsychiatric disorders, we have a nearly fair 50-50 split in terms of male and female iPSC lines of study when it comes to these particular diseases. So we can not only try to understand the phenotypes associated with X reactivation, but we can also begin to look at genetic sex differences in these particular disorders. And additionally, as far as the age of donor at sample collection, it's pretty broad across the spectrum. We have as low as, I think, 4 and 5 and all the way up to the late 90s. So I'll highlight that in the next slide somewhere. There was also addressed a need for family controls throughout the use of IPS technology, and with that, a large focus of ours has been to acquire family cohorts. And so we have a substantial portion of our resource where there is at least one or multiple family controls. And so we have 48 families where there's at least 3 or more individuals. And this could be a proband and 2 parents, 2 twins with a disorder and a parent. There's a large mixture that allows us to answer a lot of interesting questions. We have 4 families with more than 5 individuals, and we have 1 family with 9. So we have a very large subset of cell lines we can use. And one of the interesting things about having information like this is that you can find a story in the data. And one particular story that I want to highlight, and there are several of them, is a pair of parents that are 93 and 92 who donated a sample at this age with their 61-year-old daughter who has schizophrenia. And so I think it goes to show there's sort of no age limit to being able to make a meaningful contribution to sort of combat a disorder that's obviously affected this family for 6 decades. And there are 4 or 5 more examples of something just like this. So I think it's always interesting to try to look through the data and find an interesting story that can make what you're doing meaningful. So we're going to take a short transition to looking at the actual protocols and procedures that our cell lines undergo when they come in-house. So one of the things we're going to do to reduce variance is ensure that every cell line undergoes the exact same procedures and protocols. And if they don't, we keep detailed records of where they might deviate and why they might have deviated. So each cell line just goes through this similar protocol where they undergo essentially 2 passages post-arrival before they are banked. During this expansion, we do keep track, we record a growth rate metric to try to identify cell behaviors and maybe ask interesting questions about why some behave differently than others. They're then submitted to the genomics platform for sequencing. And I'll talk a little about the sequencing in a minute. So in order to submit for the genomics platform, at Bank, we collect a cell pellet where we extract DNA. We then request a sample kit from the GP, submit, and then we just sort of walk it over to 320 Charles as extracted DNA. And here we submit our samples for 2 different arrays. First, the sample will go through the global screening array, which is a SNP genotyping array. So we can identify abnormalities and other things I'll talk about in a minute, as well as each line undergoes 30x whole genome sequencing. So this is a part of that genetic resource we want to offer the community. So you can look at the genetics and find out which samples are particularly interesting for your research. Each unique sample is whole genome sequenced once, but there might be multiple clones from each donor, and each clone will undergo a global SNP array screen. This allows us to sort of address some of the questions about just having large amounts of iPSC data to determine how they behave in culture and ask questions about their stability over time. Okay, so we'll have one sample who has multiple passage numbers, and we can sort of compare their genomes and their behaviors with that. It goes through a few short logistical steps. There's a data delivery to Terra. Then we move the data on to the premises servers where we do a file conversion and put them through our CNV pipeline analysis. We do this for a few different reasons. First thing we want to do is validate expected genotypes. So if there's a particular variant of interest for which this patient was selected for, we want to make sure that it's also present in the stem cells to make sure that it didn't disappear. And so that way, if we're actually studying phenotypes, the deletion is present in the cells. Additionally, as Ralda had mentioned about trisomies and chromosomal alterations that might occur in culture, we want to identify any sort of culture-acquired variation. So here is a particular example of a trisomy, a triplication of the P arm of chromosome 12. Okay, this is lethal if you were a human, but if you're a stem cell, you replicate much more quickly. Okay, so what we want to do is ensure that we either inform collaborators of this mutation, or we just exclude it from essentially the availability in the resource. But if you're interested in studying things like the gene NANOG, which is an iPSC proliferation marker, it exists here. So it could be useful for that purpose. We also inspect the visual quality of the iPSC lines and make notes and detailed records of if they particularly don't behave well, and then we can sort of manipulate them to try to improve their quality. So what we're looking for is nice round cells, sort of nothing growing in between these little crevices. And you can see one of high quality and low quality. It might be hard to see, but there's a lot of post-mitotic cells floating around in here. Okay, this is not a cell line we would want to bank and send to a collaborator, because then you're going to have to deal with trying to clean this up and ensuring that whatever phenotype you might see could be a result of the differentiation and not the innate biology of the stem cell. So we do that a few different ways. We will pick the colonies so you can remove the center of these clean colonies and re-expand those to ensure that the post-mitotic cells don't get put into the new dish. Otherwise, you can try to add small molecules such as XAV, which promotes pluripotency, and try to sort of wean out those differentiated cells. Sorry if I blinded somebody. I had mentioned earlier that we do calculate growth rates. This way we can just have a little overview of cell behaviors in culture. And if we see differences in cell behavior, are there questions we can sort of ask right away about why they might behave differently? In fact, if we look at cell lines that have somatic and culture-acquired mutations, you can see they already have a significant growth advantage. And so we can sort of pull these out immediately as a particular phenotype associated with whatever event we've identified and recognized. And whether it's a known sort of tumor mutation, for which is why the line might have been selected, or something that has come later on that wasn't present in the patient. Addition to sort of cell behavior and visually inspecting the cell lines, we want to run a concordance analysis. This is extremely important because we want to make sure that whoever we put into our pipeline is whoever comes out of our pipeline. And in some cases, thanks to work with Dan and Dia, again at Stephen McCarroll's lab, we can identify if sample swaps have occurred or perhaps a mislabeling, and we can correct these. Because this is just going to be pivotal for anything downstream and causes a lot of problems if they do not line up. So that's a blanket overview of, yeah.
Let's say you're trying to make a line from somebody who's been identified as genetically interesting and it flunks your QC metrics. Do you keep the original material for rederivation? How do you proceed?
So,
yeah, often we do if we have access to even more. We have some patients with specific mutations we're interested in.
22q, for example.
Right, exactly. We have had some of these fail, and so we try to reprogram them again. And so if they fail again, then we can recontact the patients and get another sample. We've had cases like that, and then it either works or doesn't work. So it depends on how badly we want.
Yeah, no, that
samples.
absolutely. Sometimes you just desperately need that sample.
Definitely.
Okay, thanks.
yeah.
Maybe I can ask a question while the microphone is close.
Yes.
You said that you genome sequenced every line. How searchable is that information? So let's say I have a mutation that I want to study, and I want to know whether you have a line harboring that mutation. Can I just go to a website and just find it?
So there's no website yet. You would reach out to sort of Anna Newman and Dia and Stephen McCarroll's lab, who sort of control that genetic information, and start a conversation around that.
Wait. So I go to someone, and then how do they find out?
Yeah. Sorry.
Yeah, or maybe Dia can elaborate. But so everything now lives in the cloud, and the main bottleneck there is who has access to what data. So we first have to clear the compliance aspect, and once that is cleared, and so we've given access to whole genome sequencing data or genotyping data to collaborators, or we have, if the compliance issue is a little bit iffy, we have been able with Dia's help, so people who do have permission and access to that data, they can collaborate with others who are interested in these mutations and highlight which cell lines do or do not have the mutation. Or if once the compliance aspect has been cleared, then we can hand over or point these people to where the data lives on the cloud, and then they can manipulate it as they wish. Yeah.
So I guess I don't really understand that. So the compliance issue, I mean, I just want to know whether or not you have a cell line that has a mutation that I'm interested in case I am thinking of doing an experiment. So just the querying of that requires some kind of compliance test.
Yeah.
And then how long would it take for me to find that information?
So
on average, you know?
So again, it depends if you're okay with looping in like someone from our team. So like Gia Ghosh, for example, has done that for Beth Stevens' group and for many other collaborators that are outside the Broad, where it would be too complicated to give them access to the data. Some people like to look themselves via very sophisticated methods that they want to develop themselves. They like to query our data set, and so this is when we really would like to give them access to the data and go. So I think with Anna and Genevieve's help now, we have really kind of framework established for this, and Anna would be able to speak more about the time. But we have in-house now someone who's able, if it's a standard mutation that we want to pull out from the data, then I think it can be done very quickly, right? Yeah, in days. Perhaps you can.
Sure. I think to follow up on what Ralda just said, we have actually built a portal to this effect for our human embryonic stem cell lines, which can be accessed once you get permissions from dbGaP. And it exactly lays out a query-based method to inquire which mutations are in which cell lines and to be able to select those cell lines. We don't have what maybe Morgan is alluding to, which is just a very simple basic query of specific mutations where the user doesn't have to have direct access to the data, but there's an output which provides a list of cell lines, but that I think is a great idea, which we might want to consider as a resource for the community. Yeah.
Okay, so now I'm going to talk about a few of the ongoing collaborations and projects that we're working with, both within our group and with collaborators here at the Stanley Center, and ways this sort of resource is being currently used today. So far to date, we have distributed over 800 iPSC lines to collaborators both here at the Stanley Center, Harvard Medical School, and the Stem Cell Regenerative Biology Department at Harvard. And these researchers are using these to do a few different things. First, develop novel technologies that might enable us to link human genetics to functional biology, as well as investigate molecular phenotypes that are associated with the spectrum of rare and common variants of particular interest here at the Stanley Center in neuropsychiatric disorders. And so I'm going to go just highlight a few of the projects that we're working on. The first one is a variant to function funded initiative with Shomo Rashadri and Ann Carpenter's group. And the aim here is to define the genetic architecture of cellular traits and phenotypes. And so we're utilizing the cell painting assay, which was developed in Ann Carpenter's group, which defines morphological profiles that comprise about 2,500 different cellular traits. And that generates a distinct profile for an individual cell line. And so our goals here are to use this assay on 500 of our iPSC lines. And then once we have these distinct profiles for each of the 500 lines, we can sort of use fine mapping to identify loci that are mediating particular traits of interest. So this project is currently underway and is moving along quite steadily. The next project we're working with is something many of you may have heard about from Jana Mitchell and Jim Namesh and Kevin Egan and Stephen McCarroll's lab, is to fuel the development of these village-in-a-dish experimental systems, alternatively known as sort of jopulation and census sequencing. And these are aimed to address some of the pitfalls that Ralda talked about the cost, variance, and reproducibility of stem cell models. And so the traditional paradigm is generally to grow a single genetic background into a single culture dish. You perform your assays, then you compare the data, but there's a lot of variation that exists just between these two. And so to overcome this, we pull hundreds of people together and assay them in the same vessel and context. And this allows us to have villages that are genetically diverse enough to get cellular EQTLs, to nominate alleles that mediate gene transcription, as well as map genetic influences on cellular phenotypes. So this is the census-seq that some of you may have seen previously here. Sort of using this similar system, we've started collaboration with Alina Chan and Ben Deverman to explore how human genetic variation mediates the efficacy of AAV transduction. And this could hopefully allow us to understand how human genotypes do influence our response to AGV transduction, as well as, if there is a difference, identify which variants are responsible for that influence. And in the future, things like this are going to be very useful in hopefully informing patient selection in clinical trials and gene therapies, as well as allowing us to better develop and engineer AAVs that are effective across a broader range of human genotypes. We're also working with Sami Farhi, Bernardo Sabatini, Fei Chen, and Tongtong Zhao and his group to work on an optical profiling platform where we can perform all optical electrophysiology using an OptiPatch construct developed in Adam Cohen's lab at Harvard to develop functional profiles of stem cell-derived neurons in a dish, and then use in situ SNP sequencing to identify the cell lines based on their spatial location to look at interactions between different neurons, whether it's a disease and non-disease, or just different genetic backgrounds, and see how their electrophysiology might differ. Another project that you have all likely heard about is one led by Ralda Nehme and Olli Pietalainen with Kevin Egan, investigating the impact of the 22q11 deletion on molecular phenotypes across neuronal development. So this particular deletion is the most penetrant associated with schizophrenia, but it's also implicated in many other neuropsychiatric disorders like ASD and intellectual disability. And the biobank has really helped to fuel this because they originally had a small cohort where they did their power calculation, the one that Ralda had showed previously, and realized they needed to really scale this up in order to find, to be powered enough to find differences in their phenotypes. And so the repository was able to gain a lot of samples that carry this mutation of diverse genetic backgrounds. And their goals with this study is to differentiate these cell lines, and then at various stages, the pluripotent stem cell stage, neuroprogenitor stage, and the stem cell derived neuronal stage, perform molecular assays like RNA sequencing, proteomics, and electrophysiology to identify where pathological phenotypes might converge or diverge at different stages in neuronal development. Finally, we're working with Julia Genovese and Stephen McCarroll to generate isogenic wild type and deletion lines using clonal selection of lines that have been identified to have mosaic chromosomal alterations. So what these are is Julia had developed a pipeline to identify subsets of cells that might have particular events amongst the population. And so our aim here is to single cell select these so that we can isolate lines that have no event or lines that have a particular event that we're interested in, and then expand and bank them so that we can, as opposed to using CRISPR, we can let nature sort of generate our isogenic lines for us. And so this is a project that we're very excited about and is currently underway. So none of this at all is possible without a very large group of people here at the Stanley Center and all of our sample providing partners. Everybody plays a very important role, not just in creating the resource, its organization, as well as being inspired to use the resource and sort of fuel our further efforts to do that. And even more so, of course, all of our donors and their families that are willing to devote literally a piece of them to our scientific efforts. So with that, we'll take questions.
Yes, so how do we initiate collaborations? Or if we, for example, find a cell line that has a mutation we are interested in, how do we request it and what can we do with it? Like, can we perform all experiments we want? Can we publish? Can we use it?
So as we already said, I think once we're working to have broad use and shareability of the lines and data, depending on which line you're interested in and which collection. We might be, all clear for that, or we might be a little bit further back in the process. So it could take, it could be just as simple as us handing you a vial, or it could be as, complicated as, okay, we need to add you to some protocols, which might take a few weeks or perhaps a couple of months, and then hand you the vial. You can contact me or Anna Newman, or we also have a Stanley Center Biobank at scbb@broadinstitute.org, which you can email to request the lines. So, yeah.
It seems like it's a really fantastic resource, and thank you for describing it. I'm wondering, other than, you mentioned you have diagnoses for many of the individuals who have given their stem cells. I'm wondering if there's any effort to collect additional clinical or kind of behavioral data that gets a little more fine-grained than particular diagnosis, if there's kind of a systematic way of collecting that across the sites that you're getting these samples from.
Yeah, so right now we have basically a big collaboration that's funded by NIMH with the California Institute of Regenerative Medicine, and there they have a big catalog of different diagnoses. So if you're interested, and I think we've worked a little bit with Beth Stevens' group, that they were interested in specific Alzheimer's lines or other diagnoses, and so they were able to browse through that and select what they want. Or we are also, the other kind of arm of that is that we're trying to tap into the existing genetic collection. As we've discussed, human genetic collections are trying to diversify, and we want to really tap into these resources so that we can also diversify our iPSC collections. So if there's already a path that's open and you are interested in any of these, or if you have, again, specific interests, we can work with you. We can sit down together and say, think about what you're interested in and how can we get these cell lines. I mean, we're always open to
I
more
was thinking
collaboration.
of things like for the schizophrenia or bipolar patients' age of onset. There's evidence that those.
Yeah, so we have early and then late. So we have the stratification as well. Yeah.
I mean, one issue is the source. We can't have information that no one ever asked.
Totally. Yeah.
And I'm hoping that as our new phenotyping tools roll out, Effect, Guide, Jake, and so on, provide them, that the cohorts, I mean, in Finland, I think there was much deeper phenotyping and there's donation of cells. And I think right now, honestly, you just have to use one of the newer cohorts for your source of cells. Now, if you have a really rare, ultra-rare mutation, it just may not be possible. Probably the older cohorts, people were not actually consented to be recontacted. So I mean, we inherited a lot of those, but we've learned along the way.
Yeah, exactly.
Yeah.
Yeah.
Morgan and then I.
I really enjoyed your introduction explaining all of the issues and especially the issue of the great degree of variance that's involved, variance CE, not TS, and how you're going about it. But it sounds like, at least when in the paper that you referred to, that vast majority of variance was due to noise. I mean, maybe not noise, but not due to genetic variation. So how are you currently trying to strategize? Or how should we all think about strategizing multiple lines from the same person to figure out what kind of things will let you actually see true genetic noise as opposed to genetic signal versus?
Yeah, so there have been actually many studies that show that always pays off much more to look at different people rather than different clones of the same person. That's always going to be much more rewarding. It's better to put your money in that bucket. Like I mentioned, just trying as much as your budget or collaborations can take you, try to get many more biological replicates, many more number of people, different people, be it controls, being people that carry the specific genetic variant or diagnosis you're interested in. That's always going to be a good way. And then just choosing, I think that the protocol that you follow for making your cell types of interest, that's also going to be very important, as some are much more noisy than others. If you might have noticed that induced pluripotent stem cells in that same chart, they have much less variation as opposed to the differentiated cell types. So the starting point is actually not very variant. It's as good or even better than tissues, right? But then when you get to the differentiated cell types, then that's where the variance occurs, yeah.
Yeah, I guess I have a couple of questions / comments and related to variance as well. So, and by the way, this might be an unfair question because you were not the designer of the experiment. But in the experiment where you have a village of different iPS cells, and you're looking at different activities or whatever, by imaging or physiology, and then you're trying to associate that with a genotype. To me, that just seems unlikely to give you a lot of genetic signal, because when you culture neurons in a dish, their activity is more related to what cells they're contacting, which part of the dish they're in, and so on, which is sort of random, rather than their genotype. That's
with the electrophysiology.
Or morphology or whatever. I mean, the variance of that assay, right? I mean, in the end, you've got to find a good assay. The variance of electrophysiological activity within a dish, even if you just plate from one individual or even from one animal, is huge just within the dish, and so it's going to be difficult to associate it with genotype
Yeah.
if they're mixed all together randomly. Yeah.
So if I may take that, so this is a very good point and one that we're always thinking about. And the way we're approaching that is to really design our experiment in a way where we have pools or just even pure populations of individuals carrying a specific genetic variant so that we can also study the cell non-autonomous effects. But there are other things like intrinsic excitability, for example, that we can study within a pool. So there will be some phenotypes, if you wish, that we can study in this pooled format, and that where we can be powered enough with a very reasonable budget to look at many, many hundreds, thousands of people, and we can make discoveries that are not unlimited, of course. They are only limited to the non-cell autonomous effect, to the cell autonomous effect, rather than non-autonomous effects. So we just have to design, and then we can pull together other populations of interest to study the other effects. Yeah.
No, I was just, I mean, Morgan asked the critical question, but I think to the prior comment, I mean, unfortunately, the literature is full of publications comparing, N of 3 with schizophrenia or bipolar with 3 controls, published in high-profile journals, and it's making the field disreputable. I think that it's that what you guys are doing, trying to reduce technical variation. So I would second Morgan's concerns. I think that what you're doing is really very, very interesting, and there have been some promising results. I don't know if Lee Rubin is here, but I think the SMN experiments were telling. So progress is being made, but really critically important.
Yeah, thank you both for the great presentation. So I'm curious, out of the 670 lines that the Stanley Center collection currently has, you've talked a little bit about compliance on the side of the data, but what about compliance for actually being able to use the different cells, and is it different versus using the cells and sharing them amongst collaborators versus the data?
Again, so it's usually similar, and Anna Newman might be able to speak more to that. It's similar as far as data or cells for material. It's similar per cohort. So cell lines that we get from Finland will have their own regulatory frameworks and compliance aspects. Cell lines that we get from the California Institute of Regenerative Medicines will have, so it's all by cohort usually, yeah.
So for the patient-derived lines, the European cohorts, we are building a repository protocol with partners. That will allow us to, based on each single consent group, develop a SOP with the sample provider in Finland, in Sweden, or somewhere more locally, for how people can finally request these lines and use them in their lab. That mechanism has not been set up yet. We're working on it every day. And have been since Genevieve and I took over the project around March in terms of compliance. So for the patient-derived lines, there's a path forward. It involves a lot of conversations with legal groups across the world, compliance groups, and there's a path. We can't really say how much time that will take. In terms of the CERM iPSCs that we purchase under a licensing agreement, we have talks going on about how to make individual lines available, at least with the Stanley Center and Broad, and potentially being able to provide pools at a larger level, but we're in early talks about being able to make this resource available. Those are kind of the tentative directions that we're going in. For now, collaborating directly with Kevin is kind of the only realm of possibility right now, and as many of you know in the room, I'm working with you each very carefully. There's dozens of consents and MTAs and collaboration agreements to review as a part of this large collection that boasts over nearly 700 lines, which is, it seems like we're close, but there's definitely a lot of logistics in the way, and it's not, these lines have come from a lot of different sources.
The term strategy is for us all to die of old age, then we stop pestering.
We'll see how the next year turns out, and we'll definitely keep you all posted.
Thank you very much. Thank you.